So far, I've described two types of crosses performed by Gregor Mendel, the monohybrid cross and the dihybrid cross. So again, his observations of inheritance patterns for individual traits in these two types of crosses allowed him to derive fundamental concepts about heredity, about inheritance. So those conclusions have to do with this concept of diploidy, the concept of an allelic relationship, and more specifically in his case, dominance and recessiveness for the allelic alternatives. He also proposed this law of segregation, again, which describes a characteristic of meiosis. And then lastly, from the dihybrid crosses, he was able to derive this law of independent assortment. Again, a law that is based on a characteristic of meiosis, the independent assortment that occurs for non-homologous chromosomes during meiosis. So you can continue this sort of analysis. You can increase the complexity of the crosses by adding in additional traits. And so here's an example of a trihybrid cross. So you can choose three different traits, identify two parents that are pure breeding for contrasting forms of expression for those two traits, hybridize them to generate an F1 hybrid, and then either self or intercross the F1 hybrids to generate a segregating F2 population, and then determine if there's any particular pattern of inheritance for those three traits within that single set of crosses. So, the classic Mendelian approach. So here's an example of that. Here's two parents. We'll just talk about this generally. Three different traits, the A, B, and C trait. There's a dominant form that's being represented in this parent here, and a recessive form of expression for those three traits exhibited in the other parent. Those two parents, pure breeding parents, are hybridized, generating an F1 hybrid. And again, more specifically, that individual is going to be phenotypically expressing the three dominant characters for the three traits, and also going to be genetically heterozygous for all three genes. So if we were then to continue the analysis into the F2 population, in other words, if we wanted to predict the outcome among the F2 progeny, both in terms of genotypic and phenotypic segregation ratios, how would we do that? Well, what you would do is you would use the Punnett square, just like we did previously for the monohybrid and dihybrid crosses. So in order to perform the Punnett square analysis, you have to determine the gametic types that an individual can produce. And then you build your boxed array, and then you determine genotypic and phenotypic outcomes among the progeny. So in this case, if you're heterozygous for three genes, and again, we're assuming independent assortment, the Mendelian condition of independent assortment, that's all we know for now, you should be able to determine that these would be the 8 different gametic types that could result from a triply heterozygous individual. Okay, so if an individual can produce 8 different gametic types, and that's true for both parents, then your Punnett square to predict the outcomes in the F2 population would be an 8 by 8 boxed array. So you have 64 boxes to fill in. And then to summarize all of that information in terms of a genotypic and a phenotypic segregation ratio among the progeny in that F2 population. So you can do that. I mean, it's perfectly fine, and it'll give you the correct answer, correct information, and so on. But there's actually an easier way to do it, because when you're dealing with an 8 by 8 boxed array, it becomes a little bit time consuming and cumbersome to handle all of that analysis. So the alternative is what we call a branch diagram, and I referred to this in a previous lecture. So the idea is that as long as you have independent assortment, you can apply that product law of probability within this tool, this analysis that we call a branch diagram. So again, for our particular example, if you look at each gene individually, in other words, analyze the outcome of the cross in that F2 population for each gene individually, what would we predict phenotypically? We would predict that for the A trait, is 3/4 of the F2 progeny would be dominant for A, and 1/4 would be recessive. What about if we just analyze the B trait? So again, we'd expect a 3-to-1 segregation ratio for the B trait. And then again, if we concentrated exclusively on the C trait, we'd expect the same thing. Okay, so we're analyzing the F2 population for each trait individually, but we don't want to do that. We want to look at them in combination. So say we wanted to look at A and B in combination. So what proportion of the F2 progeny would be dominant for A and dominant for B? Well, that would be 3/4 times 3/4, right? So that would be dominant for A and dominant for B. And now this is true because of independent assortment. So we talked a little bit about this previously. So overall, you'd expect 9/16 of the population to be dominant for A and B. What about for dominant A and recessive B? Well, it would be 3/4 times 1/4. So 3/16 of that F2 population would exhibit that particular phenotypic combination. And then you fill in the rest, recessive A and dominant B, recessive A and recessive B. And the overall result, this is where we're analyzing the results here, just, that F2 population as a dihybrid. You would predict 9/16 it would be dominant, 3/16 it would be dominant, recessive, 3/16 it would be recessive, dominant, and 1/16 it would be recessive, recessive. So that's the dihybrid, the 9 to 3 to 3 to 1 phenotypic segregation ratio that we determined previously using a Punnett square analysis. Okay, so now we're incorporating a third gene. And again, as long as that gene is assorting independently of the other two, then you continue to add branches to this diagram, branch off of each of these endpoints. Okay, so again, what proportion of the F2 progeny would we expect to be dominant for all three traits? Dominant for A, dominant for B, and dominant for C. It'd be 3/4 times 3/4 times 3/4, which is 27/64 of the population. What proportion would we expect to be dominant for A, dominant for B, and recessive for C? Well, that would be 3/4 times 3/4 times 1/4, which is 9/64 of the population. So you continue to branch off for all, or multiply through all of the 8 terminal branches of this branch diagram to come up with an overall prediction for a phenotypic segregation ratio among the F2 progeny of a trihybrid cross to be 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. Okay, so again, this is the exact same information you would get out of that Punnett square that was an 8x8 boxed array. You would derive the exact same information. But hopefully, if you understand how to apply this branch diagram, this strategy is a little bit more straightforward, and you get to your answer much more quickly and more easily. So you can do the same thing for genotypic outcomes in that F2 population of that trihybrid cross. So again, if you look at each gene individually, how would we expect them to segregate genotypically in that F2 population? Well, it would be 1 to 2 to 1. So 1/4, if we're looking at the A trait, 1/4 should be homozygous dominant A, 2/4 should be heterozygous A, and 1/4 should be homozygous recessive A. Okay, and that's what you would expect for a monohybrid cross. What about a dihybrid cross? Well, again, we're assuming independent assortment. We're assuming Mendelian conditions. So this is where you can use the branch diagram. So again, of those that are homozygous dominant A, we would predict that 1/4 of those would be homozygous dominant B, 2/4 would be heterozygous, and 1/4 would be homozygous recessive. So you do the same thing here. Of those that are heterozygous for A, 1/4 should be homozygous dominant B, 2/4 should be heterozygous, and 1/4 should be homozygous recessive. And then you do the same branching down here as well. So if you multiply through, you see there's 9 branches to that branch diagram. That's the genotypic segregation. That will give you the genotypic segregation ratio for the F2 progeny of a dihybrid cross. Okay, and so we've actually seen that previously. Okay, so now we're analyzing a trihybrid. We have a third independently assorting gene. So you just continue to branch off from each of these endpoints. So again, of those that are dominant for A, homozygous dominant for A, and homozygous dominant for B, 1/4 should be homozygous dominant for C. 2/4 should be heterozygous for C, and 1/4 should be homozygous recessive for C. So you do all of these branches, you end up with a total of 27 endpoints. These are the genotypes, 27 different genotypes in that F2 population in a very specific predictable ratio. 1:1, 2:4:2, 1:1, 2:4:2, 4:8:4, and so on. So this again is the exact same information you would get out of a Punnett square. However, you derive the information in a much more straightforward way. And this diagram may look complicated, but if you understand the basic logic behind what you're doing in this branch diagram, it's actually fairly straightforward. Okay, so what if you were to continue the analysis where, say you wanted to perform a cross where you had 2 pure breeding parental types that contrasted in their form of expression for 4 traits or 5 traits or 6 traits, and you wanted to do a similar type of analysis. So things get a little bit more complicated, even with a branch diagram. But what I want to point out here is, first of all, is that there's predictable patterns in terms of the outcomes resulting from these different types of crosses. And so what do I mean by that? So for instance, if we say you wanted to, you had a tetrahybrid cross and you wanted to determine, you generated an F1 tetrahybrid heterozygous for genes A, B, C, and D. You wanted to, say, do a Punnett square analysis. So you have to determine the gametic types that individual can produce. So how would you do that? Okay, well, what this tells you here for a tetrahybrid is that you should be able to determine 16 different gametic types. So actually your Punnett square would be a 16 by 16 boxed array. Okay, remember for the trihybrid, it was 8. Remember if you're heterozygous for A, B, and C, as I just showed you previously, there's 8 different gametic types that can result from independent assortment. For the dihybrid, again, if you go back to the dihybrid analysis that we've discussed, there were 4 different gametic types that result from independent assortment. The Punnett square was a 4 by 4 boxed array. And for the monohybrid, say you're just heterozygous for A, well, then it's the gametic types are going to be 2, so it'd be a 2 by 2 boxed array. So there's actually a pattern here. The monohybrid, where you're heterozygous for a single gene, 2 different gametic types that individual can produce. If you're a dihybrid, 4, trihybrid, 8, tetrahybrid, 16. So there's an exponential increase. So the general formula to use to predict different gametic types is 2 raised to the nth power here, where n is equal to the number of heterozygous gene pairs or genes within that organism's genotype. So if you're heterozygous for 10 different genes that are all assorting independently of each other, the number of different gametic types that you can produce is 2 raised to the 10th power. Okay, so there's other formulas that allow you to predict the number of different genotypic types that can result among the F2 progeny when you self the F1 hybrid. So again, for the monohybrid, if you're heterozygous for A and you self that individual to produce F2 progeny. How many different genotypic types did we see in that monohybrid cross? It was 3. What about the dihybrid? The dihybrid was 9. That's what I just showed you here. You know, the dihybrid, 9 different genotypes that occur in that F2 population. What about the trihybrid? Well, that's again what I'm showing you here. It's 27. So 3, 9, 27. What if you were heterozygous for 4 genes and they were all assorting independently? It would be 81. Okay, so there's a general formula because this is an exponential increase. Again, the formula is 3 raised to the nth power, again, where n is equal to the number of heterozygous gene pairs in the genotype. So again, if you were heterozygous for 10 genes, all assorting independently of each other, the number of different genotypes that you would predict to occur within that F2 population would be 3 raised to the 10th power. And lastly, there's one other formula here. You can predict the number of different phenotypes that will occur among the F2 progeny, again, assuming an allelic relationship of dominance and recessiveness. That's all that we know to this point, you know, the Mendelian condition of dominance and recessiveness. So how many different phenotypes did we see in the F2 population of the monohybrid cross? It was 2, right? You're either dominant or recessive, 3/4 dominant, 1/4 recessive. What about the dihybrid? Well, there were 4 phenotypic types. You're either dominant, 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 recessive, recessive, dominant, or recessive, recessive, you know, in a ratio of 9 to 3 to 3 to 1. Okay, what about the trihybrid? Well, the trihybrid, there's 8 different phenotypes. So that's what I showed you here. A total of 8 different phenotypes in that F2 progeny, or among the F2 progeny of that trihybrid cross. And again, more specifically, in a ratio of 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. Again, so there's a general pattern. So what about the tetrahybrid? It would be 16. So again, this is an exponential increase. So the formula that predicts the number of phenotypes in that F2 population, assuming dominant recessive alleles and independent assortment, it would be 2 raised to the nth power, where n again is equal to the number of heterozygous genes within that particular genotype. So 2 raised, if you had 10 different genes, it would be 2 raised to the 10th power with regard to the total number of different phenotypes you'd observe among the F2 progeny. Okay, so that's the basis for the work that was performed by Gregor Mendel. So he did his experiments. He derived his conclusions from those observations, from those experimental results, and he went about. He published the information. He went around to various seminars and conferences to try to spread what he had learned to others. And the problem was is that very few other biologists at the time really understood what he had done, or certainly the significance of his work. And so he was pretty much ignored. One of the problems was, the interpretation's a little bit convoluted, where he's interpreting these ratios. He's talking about the concept of segregation and independent assortment, again, without any understanding of the nature of chromosomes, or an understanding of the process of meiosis and so on. So, his conclusions were pretty much ignored for several decades. It wasn't until around 1900 that eventually his work was repeated by 3 other scientists. And so it was at that time that everybody came to understand the significance of what Mendel had done many years earlier. Okay, so what happened what allowed for a better understanding, again, between 1865 and 1900, is that chromosomes were discovered, and their patterns of segregation through mitotic and meiotic cell divisions were characterized. And so it was an understanding of those chromosomes and chromosome segregation patterns that allowed for sort of the unification of that understanding with the work of Mendel. Knowing that, certainly what we know now is that genes reside on those chromosomes. So it allowed for an easier way to understand what Mendel had been proposing previously without any understanding of chromosomes. Okay, so what this led to, again, from the early 1900s and for many decades, I mean, it certainly continues even to the present day, but the main focus for geneticists at the time were to essentially repeat Mendel's work with an organism of choice. So again, with an understanding of the basic underlying principles of heredity, you could apply them in breeding programs for both plants and animals. And again, the focus there would be on agronomically important food crops. So that's what the scientists did at the time. And they focused on all kinds of farm animals, doing genetics of different agronomically important traits in those animals so that they could incorporate that genetic understanding into a breeding program. They did the same thing with plant-based food crops, things like corn, wheat, and many other important food crops in the early 1900s. So in addition to that, with the birth of this new field of biology, is that in the laboratory, geneticists, the researchers, were also doing genetics with organisms that they could develop as a model system, a model organism for performing, genetic experiments. So focus on performing genetic analysis for various traits in the mouse, in the fruit fly, and many other organisms as well. Okay, so there was a problem that arose when all of these different investigators started to generate all of this data. So the idea is that what you try to do is generate a segregating population, say an F2 population, and if it approximates a Mendelian ratio, again, Mendel observed very characteristic ratios, in his F2 populations, as we've described, either a 3-to-1 phenotypically, or 9 to 3 to 3 to 1, or 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1, and so on. So what you would do is you generate those F2 populations and see if you could regenerate a Mendelian segregation ratio. And if you could, then you could build a genetic model based on that information. You'd have single genes controlling single traits, dominant recessive alleles, and independent assortment. Okay, so this is what everybody was doing at the time with their organism of choice for chosen traits that they wanted to analyze. Okay, so the problem is that when they generated those segregating F2 populations, more often than not, they would approximate a Mendelian ratio, but they would not be an exact Mendelian ratio. So the question is that if you don't generate an exact Mendelian ratio, say an exact 3-to-1 for a monohybrid type cross, how can you conclude that that phenotypic segregation ratio adheres to Mendelian conditions? In other words, how can you propose a genetic model based on that data that is not an exact Mendelian segregation ratio? So what do I mean by that? So here's a couple of examples. So, I said that, developing the mouse as a model system. Okay, so here they had a pure breeding short-haired strain of the mouse and another pure breeding long-haired strain of the mouse. So they wanted to determine the genetic basis, the underlying genetic basis for hair length, that trait, that particular trait. So what do? You take the two pure breeding parents and you hybridize them. And what did they get? Well, they got all hybrid mice with short hair. So these are the F1 hybrids. So actually, based on that single observation, you can begin to build at least a tentative genetic model. The tentative genetic model in this case would be that it's a single gene trait with dominant recessive alleles and short being dominant to long. Okay, that's a very preliminary model, but at least it's a starting point. So then you try to get more information, more data to support it. So what would you do? You'd intercross the F1 mice to generate a segregating F2 population, okay, to see if you could get data that would support that model. So they did that, and this is what they saw. They saw two phenotypic classes, short and long, and they segregated in a ratio of 740 to 260, so a total population size of 1,000 in a ratio of approximately 3 to 1, but not exactly a 3 to 1. So this is the problem. So some scientists might look at that and say, well, that's close enough to a 3 to 1 that I believe that's equivalent to a Mendelian segregation ratio, and therefore the genetic model of a single gene trait with dominant recessive alleles is supported by that data. So you would build a model, your F1 and your F2 data supports a model of a single gene trait with dominant recessive alleles. Other scientists might look at that and say, well, it's not exactly a 3 to 1, so it's not Mendelian, and therefore your model is incorrect. So how do you get the two different conclusions? How do you resolve that issue? Who's right? Okay, so here's another example. So another mouse example. So, pure breeding for normal tail length and a normal coat of fur, and then a different strain that was pure breeding for short tail and absence of fur, a mouse. Okay, so what do you do? So again, to try to determine the genetics for these two traits, you generate an F1 hybrid, and this is what they saw is the F1s had normal tails and a normal coat of fur. So again, just very preliminarily, you'd build a genetic model that you have two genes controlling the two different traits. You have dominant recessive alleles at each of those two genes, and the dominant allele for tail length is normal, and the dominant allele for the coat of fur is the presence of fur, so a normal coat. Okay, so how do you then gather additional information to see if you can better support that model? Well, you generate a segregating F2 population. You intercross the F1s. And this is what they saw. They saw 4 phenotypic types: normal, 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 absence of fur, short tail, normal, and short tail, absence of fur. So 4 different phenotypic types in this specific ratio. So a total population of 1,008 mice segregating in this pattern. So that pattern approximates a 9 to 3 to 3 to 1. It's not exactly a 9 to 3 to 3 to 1, but it approximates. So again, one scientist might conclude that that's close enough to a 9 to 3 to 3 to 1 that it suggests a Mendelian genetic model, 2 genes controlling the 2 traits, dominant recessive alleles, and independent assortment. Another scientist might look at that and say, well, it's not really a 9 to 3 to 3 to 1, and therefore that data does not support your model. So who's right, or how do you resolve that conflict? So they had to come up with a way of interpreting genetic data in such a way that everyone would be in agreement. Okay, so you certainly do this to this day as well. Anytime you generate genetic data, you apply this same sort of what we call a goodness of fit test to confirm that the data that you're interpreting is supporting the particular model that you're proposing based on that data. Okay, so this is what, as I said, what we call a goodness of fit test or a chi-square analysis. And what I will point out here as well is that, your book certainly talks about this. I'm going to describe the way that you perform this analysis here as well. But in addition, in the Brightspace site, there's one of the modules, miscellaneous files. There's a PDF file within that module that gives a little bit more detail, sort of walks you through the step-by-step as to how to perform this chi-square analysis. So here's the results from the first, part A of this table. The results that I just described for the mouse example, long versus short hair. We observe a segregation ratio into two different phenotypic classes of 740 to 260. So a total population size of 1,000. So that's what we observe based on our experiment. So then from that, we proposed a model, single gene trait with dominant recessive allele. So what does the model predict? The model predicts a segregation ratio in those two phenotypic classes of 3 to 1. So 3/4 should exhibit the dominant trait and 1/4 the recessive trait. So again, for a population of 1,000, the prediction is a segregation ratio of 750 to 250. So we have our observed and we have what we predict based on the model, the expected outcome. You can do the same thing down here for the dihybrid cross, second example. So there I said the F2 progeny segregated into 4 different phenotypic classes with these specific numbers, the total population size of 1,008. So that's what we observed. So we then built a model, 2 genes controlling the 2 traits, dominant recessive alleles, and independent assortment. So that model predicts a specific outcome. And this is the prediction over here. So 567 is 9/16 of 1,008. 189 is 3/16 of 1,008. And 63 is 1/16 of 1,008. So we have our observed and we have our predicted outcomes based on the model. So now what we want to do is see if that data actually fits the model. So this is where you do the goodness of fit test or the chi-square analysis. So a chi-square, what you have to do is, the first step in this process is to propose a null hypothesis. And the null hypothesis is stated as follows, is that there's no difference between what I observe and what I predict based on my model, or what I expect based on my model. So you're saying that, this data set is essentially equivalent to what you predict based on the genetic model that you propose. Same thing up here. So that's actually what you're going to be testing with this chi-square test. So here's the formula. Chi-square is expressed in this formula as follows, is simply equal to the summation of the difference between observed and expected squared divided by the expected number. And that's a number that you calculate for each of the different phenotypic classes in your data set. So what do we mean by that? So just take this first phenotypic class of the monohybrid experiment. So this is observed 740, the expected is 750, the difference is -10. So observed minus expected is -10 for that phenotypic class. You then square that number and you get a value of 100, and then you divide by the expected number for that particular phenotypic class, which is 750. So 100 divided by 750 is 0.13. You then do the same calculation for the second phenotypic class in your data set. So again, here the difference is 10, you square it, and then you divide by 250, the expected number for that phenotypic class, and you get another number, you get 0.4. So then to get the final chi-square value for that data set is you simply add, this is a summation sign, so you simply add those two numbers together for a final chi-square value of 0.53. Okay? So you do the same thing down here for the second experiment, the dihybrid experiment. So here we have 4 phenotypic classes. For each of those 4 phenotypic classes, you take the difference between observed and expected, you square it, and then you divide by the expected number for that particular phenotypic class. And so you get 4 numbers, and then you add them all together to get a final chi-square value. In this case, it's 4.16. Okay, you can't do much with those numbers. You have to do another simple calculation. You have to calculate something that's called the degrees of freedom. So degrees of freedom is shown down here. It's simply n minus 1, where n is equal to the number of phenotypic classes in your data set. Okay, so again, for the monohybrid, there's 2, so the degrees of freedom for the first experiment is 1. And for the dihybrid cross, the n is equal to 4 different phenotypic classes, so degrees of freedom is equal to 3. Okay, so once you've calculated those 2 numbers, again, that they don't really tell you anything. What you do with those numbers is you take them to a probability table based on these chi-square numbers. And again, it's a table that you will find in any genetics textbook, or you will find in any statistics textbook. So this is this chi-square analysis is a basic statistical tool that has all different types of applications, and one of those applications is interpreting genetic data. That's how we're applying the chi-square analysis in this case. So again, what I'm showing you here is a table as well as a graphical representation of these chi-square values that can be calculated by analyzing genetic data. So more typically, you see these numbers, these chi-square values, organized in this table format. So that's really all that I'm going to describe. I'm not going to rely on the graph. So for our first experiment, what you do is you come to this table with the 2 values that we've calculated, a chi-square value and the degrees of freedom. So you can see different rows of the table represent different degrees of freedom. And the chi-square values that you've calculated are actually the numbers in the body of the table itself. So all of these numbers here. So you can see those chi-square values, again, then are organized into these different columns that represent different probability values. So that's actually what you come away with from this table. You bring a chi-square calculation, a chi-square value, you bring a degrees of freedom, and you come away from the table with a probability value. So for instance, for the first experiment, we calculated, let's go back, we calculated a chi-square value of 0.53 and a degrees of freedom of 1. So here's a degrees of freedom of 1. You go across the row until you find your chi-square value. So 0.53, it's not in the table, but it falls between these 2 columns. So between these 2 probability values, 50 to 20%. Okay, so that's what we're coming away with. So for the other experiment, for the dihybrid experiment, we calculated a chi-square value of 4.16 and a degrees of freedom of 3. So here's degrees of freedom of 3. You go across the row until you find your chi-square value. So 4.16, again, falls between these 2 columns, between these 2 probability values, 20% and 50%. So it's within a range. It's not a precise value, at least, from this table here. So somewhere between 20 to 50% probability. So what does that probability value tell you? It's actually that probability value that either allows you to fail to reject or reject your null hypothesis. Okay? So what does that mean? What that probability value means is that if your model is correct, then between 20 to 50% of the time, if I were to repeat that experiment over and over again, I would expect to see this much deviation or more between the observed and the expected values due to random chance alone. Okay, so if I were to repeat the experiment 100 times, anywhere between 20 to 50 of those experiments would exhibit this much deviation or more between the observed and the expected values due to random chance alone. Because that probability value is so high, that's a pretty high value for that probability level, you would fail to reject your null hypothesis, and you would then conclude that the data supports the model that you've proposed. Okay? So in other words, this data does support, even though it's not exactly a 3-to-1, supports a single gene model with dominant recessive alleles. This data, again, supports a genetic model of 2 genes controlling the 2 traits, dominant recessive alleles, and independent assortment. So the question is, or you should be asking, where does random chance come into the interpretation of genetic data? Well, the idea is, and then allows you to apply this chi-square test or this goodness of fit test. Well, it has to do with the fact that fertilization events are assumed to be totally random events. And so with this chance being a fundamental component of reproductive events or fertilization events, you can apply this chi-square test to interpretation of genetic data. So the question is at what probability level would you reject the model, reject the null hypothesis? Well, that value is arbitrarily set at this 5% level. So in other words, if you do the chi-square analysis and you come away from the table with a probability value that is less than 5%, meaning that only 5% of the time, if you were to repeat the experiment over and over again, only 5% or less of the time would I expect to see this much deviation between the observed and the expected, then you can conclude in that case that something other than random chance is causing the difference, causing the deviation. So in that case, you would reject the genetic model as being correct. And you have to go back and come up with a different interpretation of the data. So this is pretty much arbitrarily set. And at the very least, what you can see, the way that this table here is constructed, is that any chi-square value that falls within this dark blue region, I guess, you would conclude that your model, that the data supports your model. You fail to reject your null hypothesis. And when your chi-square value falls between, this 5% level, so in these last 2 columns of the table, the lighter blue region, you would reject the genetic model, meaning that the data does not support that model because the deviation is too great to be explainable based solely on random chance. Okay, so I'm going to wrap up this discussion for now, and we're going to then begin to discuss other problems that arose when geneticists in the early 1900s started to do, Mendelian type genetic analysis with their particular organisms of choice. What I've described here is only one problem that arose, but it was easily solvable by applying, this specific statistical tool to interpretation of the data. But there are other problems that arose as well, and we'll talk about them next time.